We're starting the conversation about the immune system today. Now, I do my best to break it down. I promise, it's deeper look into immune system than what you had before. I almost guarantee that. I try to make it a bit more exhaustive because here immunity plays kind of major role in understanding the process of infectious disease. And we're going to quickly go over various what we call the surface barriers. So there's non-specific responses. They do not involve any specific enzymes or cells or protein factors, nothing chemically specific, or biochemically specific. They are either mechanical, such as mucus, say mucus in the respiratory system, or in the gut, or in the vaginal mucosa. It will trap pathogens, and then somehow this mucus will be removed. In the respiratory system, mucus is removed by cilia that synchronously move it upwards, and then you swallow that mucus with the pathogens. In digestive system, it is just moved with a normal digestive peristalsis. Now, chemical barriers include both low pH at the different locations. Skin is slightly acidic, while vagina is more acidic due to the presence of lactobacilli species. The most acidic site, of course, is the stomach, and that explains why many microorganisms simply cannot survive there. There are some more specific chemical mechanisms, such as lysozyme, one of the most famous secreted antibacterial proteins, found in virtually every watery secretion from saliva to tears. It is even found in mucus itself. Skin produces specific proteins called dermicidins, and they look like they may be a promising candidate for the development of actually new antibacterial drugs, if they can get chemically modified to alter specificity or efficacy. Human skin may actually be a valuable source of antibiotics. There are some works showing that sebaceous secretions contain short-chain fatty acids that may have some antibacterial properties. But as we discussed before, probably the most powerful defending system in the body parts like skin or vagina or gut, microbiome. Okay? All the microorganisms, they serve as the first barrier to prevent infections, to increase competition, to restrict the nutrient availability for the potential pathogenic microorganisms. Does that make sense? That brings us to the conversation of innate immune system and the cells that mediate innate immune responses. So, innate immunity, it's already there. It exists right away. It responds extremely fast. In order to develop the innate immune response, cells, they have to recognize pathogen, but they do have to recognize pathogen as a pathogen, but they do not require any time to mature, to develop, to be selected. If pathogen is discovered, innate immune response kicks in right away. So basically no exposure is required. However, that lack of specificity, well, they're fairly non-specific, which is sort of a disadvantage. You know the expression, the jack of all trades, right? So innate immunity is kind of a jack of all trades, but the master of none. So it responds to virtually any pathogen invasion, but the response has a limited effectiveness. Most importantly, innate immune responses are usually local. They aren't systemic, so they will elicit some defense at the site of infection, for instance, but will leave other parts of the body totally unprotected. However, as we mentioned, the advantage is that this response is really rapid, kicks in first. Now, there are two types of white blood cells that respond immediately, parts of innate immune response. First type is granulocytes. Among granulocytes, we need to talk about the following four cell types. First are neutrophils. Sometimes they are called polymorphonuclear cells. This is more clinical. I'm going to use neutrophils, but if you read clinical literature or during your nursing classes, the acronym PMN comes up. That's same as the neutrophil. Polymorphonuclear refers to the multi-lobed nucleus. These are the most abundant white blood cells found in circulation, and their main target are bacteria. If a patient has clinical neutrophilia, elevated levels of neutrophils, you can quite reassuringly say that the patient has some kind of a bacterial infection. How do neutrophils fight off bacterial pathogens? First and foremost, neutrophils are phagocytic cells. They engulf and destroy pathogens. Unfortunately, or fortunately, as the result of that engulfment and destruction, neutrophils die themselves. They kind of kamikazes of white blood cells. And when they die, they release chemicals of respiratory burst, which sounds really fancy until you figure out what these chemicals are. Hydrogen peroxide and bleach. Quite literally. Hydrogen peroxide and hypochlorite. Okay? So they release those chemicals that kill bacterial cells. And on top of that, they use an incredibly elegant protective mechanism that is called nets, or neutrophil extracellular traps. When neutrophil dies, it releases its DNA in the form of long threads. These threads literally wrap around bacterial cells and affix them at the spot. Does that make sense? I find it like it's a great name because it's like a net that bacterial cells are being caught by. And there are multiple studies, there are beautiful microscopic images showing bacterial cells all wrapped with the DNA threads and tethered to the spot. But, those are three main mechanisms. Second type are eosinophils. Now, eosinophils, shown here and in this micrograph, has usually bilobed nucleus and can be stained with acidic dyes. So neutrophils can be stained with both. This is why they're called neutrophils. These are called eosinophils. So eosinophils target mainly eukaryotic pathogens, from fungi to protozoa to helminths. How would you compare the size of the cell between the bacteria, some kind of eukaryote? Which one's bigger, eukaryotic or bacterial? Eukaryotic. So phagocytosis becomes a problem, right? It's really hard to engulf and destroy something that is larger than you. So eosinophils use different tactics to repel and destroy eukaryotic pathogens. They release various toxic proteins, we call it cellular toxicity, and they also produce chemicals of respiratory burst. Put yourself in the place of eosinophil that responds to a helminthic infection. Helminth is a microscopic animal. It's large, often visible with the naked eye. And this tiny little eosinophil cannot possibly phagocytose a helminth. Does that make sense? So in response, these chemicals of respiratory burst and cytotoxic chemicals, they kind of bug the hell out of the helminth. They make the environment so toxic that helminth detaches from whatever location it is and tries to leave the body. And eosinophilia is a valuable diagnostic parameter that allows you to say, oh, okay, this patient most likely has protozoan or helminthic infection. Not always. Not every helminthic or protozoan infection will show up with eosinophilia, but many will. Does that make sense? So these cells are also found in circulation, and interestingly enough, they were implicated in allergic reactions. We don't know the exact mechanism, but it was shown that, for instance, in children with asthma, if you would take a small biopsy of the tracheal wall, you will find eosinophils migrating into lamina propria of tracheal mucosa, so compared to kids without asthma. So eosinophils are more abundant in the trachea of people with asthma. Make sense? And I'm sorry, I'm going to switch it at another screen. Okay. So now, why it makes perfect sense that eosinophils are involved in allergic responses? I believe I mentioned the hygiene hypothesis before. The idea that since we aren't exposed to various microorganisms as much when we grow up, we'll live in a pretty clean environment. For folks from developed countries, it's more likely to grow up with some kind of autoimmune disease, and allergy is a typical autoimmune disease. So here's the link. The idea. Eosinophils do not get sufficient exposure to pathogens, like, common just environmental microorganisms when we grow up. So their maturation over time is skewed, and that somehow changes their specificity. So instead of focusing their responses on helminths or protozoa, they start to attack our own tissues, releasing those chemicals causing allergy symptoms. Again, this is just a hypothesis. There's no mechanism implicated in it. But the data kind of makes sense. Make sense? The third type, or basophils, can be stained with basic dyes, also found in the circulation. And, well, I put the target here as allergy. Of course, allergy is not a target. Normally, basophils are filled with granules. You see, all these cells have multiple cytoplasmic granules. Those dots are cytoplasmic granules. And granules of a typical basophil contain a chemical called histamine. You definitely heard about it, antihistamine drugs. So histamine is released during the process of inflammation. But if basophils release histamine not during the normal inflammation, not during the infection, but during the allergic reaction, they are playing an important role in allergy. Now, mast cells, which are not on the picture, but it's basically basophils, which moved from circulation to the tissues. They contain that same histamine, and these are entirely allergy cells. When somebody has an allergic response, mast cells in the tissue degranulate. And degranulation means that granules release histamine. If that release of histamine happens in the dermis of the skin, you have rash, allergic rash, you have itching. If it happens in the mucosa of the nose, you have dilation of the blood vessels, runny nose. If all mast cells start to degranulate at the same time in all tissues around your body, you have what is called anaphylactic shock. Histamine will trigger a constriction of the blood vessels, and not blood vessels, sorry. It will trigger the constriction of the bronchioles, so you have asphyxia and massive vasodilation in the entire systemic circulation, leading to vascular shock, and that inflammatory response all over the place will increase vascular permeability. So plasma starts to seep out from the blood vessels, reducing your blood volume, leading to hypovolemic shock. That's the mechanism of anaphylactic shock, degranulation of mast cells. Does that make sense? Now, before we move on to the next set, to the agranulocytes, obviously for a cell, I'm not going to show you the pictures. Don't worry about the pictures. What I want you to know, found in circulation or in tissues, main target, main mechanisms. Am I clear? Good? Now, agranulocytes. You see there are three here, but actually there are two main types. One is monocytes and macrophages. Another type are lymphocytes. So let's talk about monocytes and macrophages first. Here on the left, you can see the monocytes clustered together in the blood. I usually compare monocytes to a Bruce Banner because when they are in the circulation, they don't really do much. But when they are activated by some kind of inflammatory response, okay, they become macrophages. So that's the moment when Bruce Banner becomes Hulk. And macrophages, these cells, they're actually the largest white blood cells. They migrate into the tissues and start to phagocytose pathogens. Macrophage, the name, suggests that these are really good phagocytic cells. Macrophage literally means big eater. Okay? So they're large, they phagocytose, and unlike neutrophils, they don't die. They just keep phagocytosing. Do you see the difference between monocytes and macrophages? Monocytes are in the circulation, macrophages are in tissues. Monocytes don't do much, macrophages actually phagocytose other cells. You clear? Now, another type of cells of the innate immune system, the second type of granulocytes, is NK or natural killer cells. Now, from the name, you can guess what these cells do to other cells. what do? They kill them. And they kill them using a very elegant mechanism. So there are actually two main mechanisms that they can use. One is a receptor-ligand interaction, which kind of shown here, but I'm going to draw my own picture. So imagine that's an infected cell. This is NK cell, right? The blue is NK cell. So it turns out NK cell carries a receptor called Fos receptor, and infected cell expresses the ligand called Fos ligand. When they come together, I really want to throw some Harry Potter Magic Wand reference here. I can't because I didn't read a single book. But basically, NK cell kind of touches the infected or cancerous cell and initiates signaling pathways that lead to apoptosis. Cell dies. Good? That makes sense. Now, a second mechanism is perforins and granzymes. Now, perforin, okay, that's infected cell again, and that's NK cell. Now, think about the name, perforin. What do you think perforin does? It's in the name, perforates, yes. So perforins are proteins that are released from NK cell. They form pores in a cytoplasmic membrane of the infected cell. And then, after the perforins, granzymes follow, and granzymes, these blue dots, they enter the infected cell and also initiate apoptosis. So you see there are two mechanisms that can stimulate apoptosis in the infected cell, either by specific receptor-ligand interaction or by releasing special proteins. Does that make sense to you? We're good? Now, how does NK cell knows which cell is bad, which cell is good? Okay? Turns out that generally speaking, your normal cells produce chemical. We're writing like, I'm jumping like a topic ahead, not a full topic, but just to explain. So all your normal cells carry a special group of proteins, called major histocompatibility complex, type 1, MHC1. You know that, huh? Major, like most important, histocompatibility. Histo means tissue, compatibility means compatible. So basically, look, it is a tongue breaker, but if you think about the idea, those are the proteins that practically label your tissues saying that's yours. Does that make sense? That's yours. So say my MHC proteins are different from Chloe's MHC proteins. This is why rejection happens during transplantation. Does that make sense? And that is why donors are selected based on their MHC compatibility, histocompatibility, tissue compatibility. Got it? So, infected cells or malignant cells do not have a proper MHC molecule. An MHC molecule serves as the inhibitor. Okay? When there is no inhibitor, when there is no molecule like this, okay, then NK cell binds to the receptor and apoptosis follows. Make sense? Good? Now NK cells work, they basically, again I'm jumping ahead, they're basically like T lymphocytes. You've heard that term before, I believe, or cytotoxic lymphocytes, but they're not really specific. What I mean by non-specific, I presume that you have an idea. Humans can produce adaptive immunity, specific immunity. This is why we use vaccines. So we can, if we get measles, we're going to be immune to measles, specifically to measles. So, and there are cells that will recognize measles antigen and nothing else. We're good? NK cells do not distinguish. What they say is that this cell is infected, this is not. What is it infected with? I don't know. But I know that it's infected, I'm going to kill it. Does that make sense? So they're kind of broad spectrum sort of things. Do you see the difference between really specific and these natural killer non-specific cells? Got it? Questions? Moving on. So, what happens in terms of innate immune response during the infection? So there's an injury. Okay? And green particles are pathogens. So when injury occurs to the tissue, infection happens, all that leads to the release of pro-inflammatory. Look, I'm going to ask, don't think, don't take it like, oh, he thinks we're stupid. Pro-inflammatory, does that make sense? They facilitate inflammation. Cytokines. Cyto refers to the cell, kine refers to kinesis, movement. So cytokines are molecules that force white blood cells to move somewhere. They basically chemoattractants. Does that make sense? So the cytokines and another group of molecules, chemokines, virtually same function. They end up in the circulation. And with the blood, they end up in the bone marrow. And in the bone marrow, these cytokines and chemokines stimulate, first and foremost, leukocytosis. When you go to the doctor and you're sick and they draw blood and they tell you, oh yeah, you have an infection, you have high white blood cell count. That's leukocytosis in response to an infection. Does that make sense? A lot of white blood cells are produced by bone marrow. These white blood cells enter the bloodstream, and eventually they will pass by that site of injury. Now, think about the situation these white blood cells are in. They are in the blood, injuries in the tissue, they're passing by it. They have to know that the injury is behind that vascular wall. What gives them that idea? Adhesion molecules. These pro-inflammatory cytokines, okay, these blue dots, interact with the receptors on the endothelial cells of the blood vessel, stimulating the synthesis of adhesion molecules. Adhesion molecules are responsible for adhesion of white blood cells. You can see the leukocyte adhering to the cell wall. They like Velcro for leukocytes. So white blood cells, when they are in the vicinity of the injury, you with me? They have better chances to stick to the vessel wall and stop there, forming a firm adhesion. Once they adhere, they start to follow the chemical gradient of cytokines. It is called positive chemotaxis. They follow it so, they committed to following it so hard that they actually penetrate through the blood vessel wall in the process that is called extravasation, or, if you want to throw some really fancy word, crossing the vessel wall is called diapedesis. Okay, you can see this white blood cells extravasating, okay, in the peripheral tissue. Does that make sense? And then they will follow that increasing concentration of cytokines and chemokines, just like Hansel and Gretel were following the trail of the breadcrumbs. Now, this inflammatory response will also facilitate the differentiation of monocytes into the macrophages. And in fact, two main white blood cell types that are recruited to the site of injury are essentially macrophages and neutrophils. They both phagocytic, and FYI, I will repeat it multiple times, but that's the first time. We have only two phagocytic, well, we have two main phagocytic types. It's the macrophage and neutrophil. Those two cell types use phagocytosis as the main defensive mechanism. Does that make sense? Now, in addition to macrophages and neutrophils crossing blood vessel wall and reaching into the site of infection, increased vascular permeability will lead to plasma leakage. That plasma will wash away the debris, the dead cells, also removing pathogens into the lymphatic system. Okay, now that plasma leakage leads to swelling, we call edema. Swelling starts to press on the sensory neurons, causing pain, and increased blood flow to the site of infection contributes to redness, and higher cellular activity at the site of infection contributes to elevated temperature, which gives us 4 signs of inflammation. In Latin, it's going to be rubor, color, tumor, and dolor. The rubor stands for redness, color stands for heat, tumor stands for swelling, and dolor stands for pain. Since we're not in a Latin class, you don't have to memorize that crap. Okay? But it kind of rhymes. Rubor, color, tumor, dolor. Finally, there are clotting factors like fibrinogen that will be converted eventually into the fibrin to trap the pathogens in place. Does that make sense? The whole story. I think that the process of leukocyte recruitment is extremely elegant. Just so logical. So, steps, people. Steps. Okay? Alright. That's pretty cool. We have just enough time to chat about it. Now, how do we know what to respond to? How do we know that this is bacterium, this is virus, this is protozoa? Pathogens have certain molecular signatures, so-called pathogen-associated molecular patterns. Now, let me ask you the question about gram-negative bacteria. They have one really important molecule in the outer membrane. We talked about it on Monday. It's some kind of a toxin. Do you remember? Gram-negative bacteria, outer membrane. What kind of toxin? It's like a whole class of them. Endotoxin. They have endotoxin. And endotoxin can be found only in gram-negatives. Clear? That's their molecular signature. If you think about the gram-positive cell wall, gram-positive microorganisms, and only gram-positive microorganisms, have teichoic acid. Okay? That's their molecular signature. Many gram-negative microorganisms have a special protein called flagellin, found in the flagellum. Another molecular signature. Viruses have sometimes single-stranded or double-stranded RNA genome. Double-stranded RNA is viral molecular signature. Fungi have, I cannot remember from top of my head, one of the chemical components of the zymogen, I think. It's found in the cell wall of a fungus. That's molecular signature. Does that make sense? So these molecular signatures are called pathogen-associated molecular patterns, or PAMPs. Now I'm going to kind of knock it in your hand. So those are molecules that are associated with pathogens, found on pathogens. Got it? Your cells have other molecules that are called pattern recognition receptors, or PRR. Pattern recognition receptors recognize those patterns. Does that make sense? Look at this cell. So, flagellin. Can you see flagellin on the right, the pink circle? That's PAMP. Understood? The receptor for flagellin is PRR. Got it? Peptidoglycan found in bacterial cell wall is PAMP. The receptor is PRR. Sorry, it's P. It should be P. Okay, does that make sense? So PAMPs are found on the pathogens, PRRs found on the host cells. Okay, now a huge class of pattern recognition receptors called toll-like receptor. And if I have a minute, I will tell you the story of the name. It's super funny. Some of the toll-like receptors are located on the plasma membrane. For instance, TLR4 can be activated by endotoxin, by lipopolysaccharide, component of gram-negative cell envelope. Make sense so far? Now, these respond to mainly extracellular pathogens, like protozoa bacteria. Some PRRs, or TLRs, like TLR3, 7, 9, they are found in the endosome, inside the cell. And they respond to the virus infection. Quick question. Why the receptors that recognize virus infection are located inside of the cell? Huh? Say again? So, well, virus infection, what does it require? Absolutely must have. A cell. A cell. Virus got to get into the cell, and that's where receptors waiting for it. Make sense? Like this phagosome, you have a virus with a nucleic acid. There you go. The receptor for nucleic acid. TLR9, as far as I remember, recognizes DNA. Does that make sense? Now, the function of pattern recognition receptors, like TLR. And that's not a complete list. Believe me, there are some cytoplasmic proteins that serve as pattern recognition receptors. If something gets in the cytoplasm that shouldn't be there, like DNA, DNA shouldn't be in the cytoplasm. If DNA is in the cytoplasm, there's going to be a receptor recognizing it. What they do, they just trigger some kind of a signaling pathway. And that will activate cellular protective response. Oh, for the sake of it, cell can just die. That's the easy way, you know? Cell dies, everything dies with it. Does that make sense? So mainly what I want you to understand is really like how this interaction is going. Say, tell you, flagellin, a part of flagellum on gram-negative microorganisms. Is that a PAMP or PRR? That's a PAMP. Or where are they found? PAMPs on the pathogen, PRR on the host cell. Now, why they called toll-like receptors? Actually, folks that discovered them got Nobel Prize several years ago. So, the first receptors like this with that function were discovered in Drosophila. So and they were called toll receptors, and we'll get why they're called toll. When same receptors were discovered in humans, those scientists found nothing better than to say, well, that's like Drosophila. I'm going to call them toll-like. And the name stuck. But Drosophila's story is showing you how scientists name things. So when the guy that discovered Drosophila toll receptor, which was not named at the time, actually made the discovery, he was German. And he looked like at the results, and he was like super excited. He said, toll! Which in German means cool. And his colleague said, well, that's the name. So those are basically cool receptors. So we're going to continue talking about the innate immunity next week.